শুরু হচ্ছে।
হ্যাঁ, ফ্লায়ারটা দেওয়া হয়ে গেছে।
আচ্ছা, ম্যাডামকে তুমি বলে দেবে, তখন ম্যাডাম বলতে আরম্ভ করবে।
হ্যাঁ, ঠিক আছে। ম্যাডাম আপনি বলতে পারেন, ইউ ক্যান স্টার্ট।
Good afternoon, everyone, and sorry for the delay due to some technical fault. I welcome to today's webinar, Immune Response to SARS-CoV-2 and its treatment strategies. SARS-CoV-2, that is COVID-19, is a new virus, produces serious health crisis throughout the world. Being a new virus, its nature is not completely known to us, and the virus, after its spreading, mutated itself many times. This nature of virus and unavailability of specific medicine or vaccine forces us to stand behind closed doors, covering ourselves with protective equipments and waiting patiently for this time of hardship to pass. Few antiviral drugs like hydroxychloroquine, remdesivir have been used in serious COVID patients in some countries with little effectiveness. Among various improved methodologies, plasma therapy has shown prominent action towards the fight against virus. Doctors and scientists throughout the world have engaged themselves in the discovery of potential vaccine against the virus antigen. We hope this effort will bring a great benefit towards the surveillance of mankind. Now we move to our main session. We are proud and honored to have an eminent scientist and immunologist, Dr. Dipyaman Ganguly from IICB Kolkata, who is our speaker today. We heartily welcome him in our webinar and extend our heartfelt thanks to be present here. We wholeheartedly acknowledge the support and inspiration providing by our principal sir, Dr. Sudip Kumar Chakraborty, for organizing this webinar. We are also grateful to our Microsoft technical team for their constant support and IQAC for collaborating with us. We welcome all the participants, faculties of different institutions in this webinar. Our principal sir send up his audio message, which will be played by our technical team within few minutes. Now, I request Dr. Chiranjit Mondol to play the audio message.
আমাদের কলেজে পেরি মোহন কলেজের ফিজিওলজি ডিপার্টমেন্ট এবং আইকিউএসির যৌথ সমন্বয়ে আজকে এই ওয়েবিনারের আয়োজন। এখানে যিনি বক্তা ডাক্তার দীপ্যমান গাঙ্গুলি, আমি তাকে আমাদের কলেজের তরফ থেকে আন্তরিক অভিনন্দন জানাচ্ছি। নমস্কার ডাক্তারবাবু।
হ্যালো।
এই বিষয়টি ডাক্তার গাঙ্গুলি বিশেষভাবে তার কাছে আমাদের আশা, ছাত্রছাত্রী যখন জানবে বা শুনবে তারাও নিশ্চয়ই বুঝবে যে এই আশা তাদের কতটা তাদের কাছে ফলপ্রসূ হলো, তারা আনন্দিত হলো। তিনি একধারে ডাক্তার, অধ্যাপক এবং গবেষক, সায়েন্টিস্ট। তো সেক্ষেত্রে এই যে প্রাসঙ্গিক আমাদের এখনকার যে সমস্যা, ভীষণ সমস্যা, প্রাসঙ্গিক বলবো না, ভীষণ সমস্যা আমাদের করোনা ভাইরাস কোভিড-19, সেই বিষয় নিয়ে তিনি বলবেন কিভাবে আমরা কোন স্ট্র্যাটেজিতে আমরা এই ভাইরাসকে পরাজিত করব, ভাইরাসের দ্বারা আক্রান্ত হলে আমরা সেই আক্রমণ থেকে বেরিয়ে আসব, কিভাবে আমাদের প্রতিরোধ শক্তিকে আমরা বাড়িয়ে তুলব, এই বিষয়গুলো নিয়ে তিনি বলবেন। আর এমন নতুন কি, আমরা তো ইউটিউবে সবসময় দেখছি, বিভিন্ন প্রিন্ট মিডিয়াতে দেখছি, আমরা আমাদের সোশ্যাল মিডিয়াতে দেখছি, একটাই আলোচনা মানুষের কাছে, কোভিড-19, কিভাবে সেখান থেকে বাঁচবো, কিভাবে আমরা ইমিউনিটি বাড়াবো। কিন্তু তাও সেসব থাকা সত্ত্বেও আজকে যিনি বলছেন তিনি ডাক্তার, গবেষক, সায়েন্টিস্ট, সরাসরি তার কাছ থেকে শোনা নিশ্চয়ই একটা আলাদা মাত্রা দেবে আমাদের এই বিষয়ের অভিজ্ঞতাকে। সেই জন্য আমরা ধৈর্য ধরে তার বক্তব্য শুনব। এই ওয়েবিনারটি আয়োজন করার জন্য আমি আবার আমাদের ফিজিওলজি ডিপার্টমেন্টকে, IQAC কে, কলেজের কর্তৃপক্ষকে, ছাত্রদের সবাইকে অভিনন্দন জানাচ্ছি। সবাই ভালো থাকুন, মন দিয়ে শুনুন। আবার ডাক্তারবাবুকে আমি অভিনন্দন এবং নমস্কার জানাই। সবাই ভালো থাকবেন, ধন্যবাদ, নমস্কার।
Now, I would request Dr. Lavanya Mondol, HOD, Department of Physiology, to introduce our speaker, Dr. Dipyaman Ganguly. Over to Lavanya Mondol.
Hello. A very good afternoon to everybody. And at the very outset, I'm really sorry for this delay due to a technical snag. Please, thank you that you have still there. Let me begin the webinar. It is a great honor for me to introduce to you our speaker of the day, Dr. Dipyaman Ganguly. Dr. Dipyaman Ganguly got his MBBS degree from the Calcutta Medical College and Hospitals in 2001. He did his PhD in biotechnology from the Indian Institute of Chemical Biology and a PhD in immunology and biomedical sciences from MD Anderson Cancer Center, Houston, USA. Currently, he is working as the principal scientist and associate professor at the Translational Research Unit of Excellence of IICB, which is a CSR Research Institute. His research interest is focused around the innate branch of innate immunity against infectious agents, against cancer, against autoimmune disorders. He has got numerous journals. His publications have been produced in various international journals. I will just name a few of the journals in which his publications are there, like the JI, PNAS, Journal of Experimental Medicine, Nature Immunology, Nature Reviews Immunology, Trends in Immunology, New England Journal of Medicine, to just name a few. He has got several patents, and he has also authored book chapters in books by publishers like Taylor and Francis and Elsevier. He is the recipient of several awards like the Career Development Award from the Department of Biotechnology, the Swan Jayanti Fellowship and the Ramanujan Fellowship from DST, to name just a few. Now, during this COVID condition, he has been the principal investigator in the only plasma therapy trial that is going on in the eastern region of the country in collaboration with the government of West Bengal. We are all eager to listen to you, Dr. Ganguly, and I welcome you once again from the Department of Physiology and the Raja Perumal College on the whole. Over to you, Dr. Ganguly.
Thank you so much. It is a privilege to be able to talk to this audience. Thanks for inviting me here. I'll just, without much delay, I'll just try to share the presentation. Do you see the presentation now? Hello?
Yes, sir. Yes, sir.
Okay.
Yes, it's visible.
So today we'll discuss about this mayhem, like international mayhem that we're going through at this point. And so I have been a small part of a few studies which is trying to figure out how it can be changed. And so we have to understand. So most of the information, so this epidemic has been a unprecedented one, right? First, it's unprecedented because it's a new virus, etc. And also, another major thing is humans were technologically very prepared to take this pandemic. Yet, 7 months have passed, we are nowhere because the virus is so contagious and so spread so fast. It was beyond our even the technological advances to contain it. And the major problem that this pandemic is posing to us is uh, because of its novelty, right? So the problem actually, so do you see the changed slide? Just confirm once. Hello? Please, anyone just confirm that you are changing, I mean, the slides are changed and you are seeing that.
I think it's still on the first slide.
This one?
Now, yeah, it has changed.
Oh, okay, yeah. So, so the major problem lies with the novelty, right? So its name is nCoV, novel coronavirus, and because when a new virus comes, the human body is not prepared to take it because it's seeing something new. But we have to remember that human civilization has been seeing pandemics for time immemorial, right? So contagious diseases started with the human society, and especially with the agricultural activity after we stopped being hunters and foragers and started settling down in different river beds, because once organized agriculture started, people had to reap the benefit of that agriculture, and so they had to be staying in one place for at least 6 months. And so that's how all the villages and then the cities and civilizations grew on river beds. And with that, the contagion started, right? Because now people were staying near to each other. If someone gets infected, another person gets contagion. And so, well, this is not a new thing. That's what I'm trying to say. So the pandemics have been there. There have been at least 10 plague pandemics all over the world. We are always seeing reference to 1918 Spanish flu. And this third planet from sun, it has seen a lot of pandemics. And actually, we are much more prepared this time, I would say, because we know much better about human biology as well as host pathogen interactions at this point. Now, this is a picture of Spanish flu almost 100 years back, more than 100 years back, and you see the similar kind of pictures that we are experiencing now. Actually, we haven't seen this kind of makeshift hospitals as of yet. In some part of the world, there have been this kind of things, and we really need to get these things fast. And I think the lockdown would have been a good opportunity for us to develop these facilities so that a large number of patients could be catered to. But what I would try to impress upon you that life never halted like this before. So during Spanish flu, this is San Francisco court being held outside the courthouse just to prevent infections from spreading. And people had been playing baseball, wearing masks. This is another pandemic in 2003, SARS pandemic. And this is a picture from Hong Kong. You can see young kids practicing ballet, wearing masks. And so life never stops, right? আমরা বাঙালিরা বলি যেটা মন্বন্তরে মরিনি আমরা মারি নিয়ে কর করি. So it's like that. So you have to you have to be rational. You have to understand information is power. You need to know what exactly is posing the threat and what exactly we can do at different phases of the pandemic to prevent from lives being lost, right? Now, there have been previous pandemics of similar viruses. All of us know about this. SARS pandemic 2003, March 2012, Ebola, which is actually ongoing for a long time. All these pandemics actually kill more people than SARS-CoV-2. So you can just look at these box plots. I mean, in SARS-CoV-2, the major problem is not the fatality. Its major problem is the sheer speed of spread. In past 6 to 7 months, we have seen 18 million cases, 18 million infections, and only 700,000 deaths. I know each death is a life lost, but still, for when you are taking a broader perspective, you need to take account of the case fatality rates and determine how to strategize your policies. Now, this case fatality, this sheer spread of infection of SARS-CoV-2, perhaps you have again seen these words in media because this has been an unprecedented pandemic in modern humanity, and naturally media has been featuring all sorts of scientific parlance in the public media, popular media. So you have heard about this basic reproductive ratio, and this is just in simple terms, this is just how many people can get infected if they meet another person who is already infected. So for most of the viruses, it ranges from 1.5 to 2.5. In case of SARS-CoV-2, there was some initial estimation from China, which said it is almost 2.2 to 2.5. But on a more recent estimation by Los Alamos National Laboratory in USA, it's almost like 5.5. So that tells you that this is infecting, the contagion is really fast. Why is it so? Because first, it's a droplet infection, aerosol infection, and second, in a normal influenza, someone who is infected remains infective for 5 to 6 days. In case of SARS-CoV-2, the duration of infectivity for an infected person is much longer. There have been, again, varying estimates because the pandemic is still ongoing, data are being collected, and there are variations in human biology. But still, it seems it ranges from almost 15 days to even 40 days. So someone, when he is infected, he or she is infected, he or she is able to spread the virus for so long. So naturally, the contagion is unprecedented for this virus. And that's why it's spread. This is the animation shows a 2-month data, how the virus spread over 2 months through all 7 continents all over the world. And just because of this interesting biology, interesting disease biology, that the patients are remaining infective for so long. But the major data that came out of China pretty early during the pandemic is there is a major age barrier for case fatalities, right? Now, all these data are case fatality rates because in the early days of pandemic, people never thought of asymptomatic carriers and asymptomatic infections. So all the fatality calculations were made from how many patients you are seeing who are reporting to the health authorities with different symptoms. And so naturally, you are actually looking at case fatality rate compared to infection mortality rate. Infection mortality rate is something else. I'll talk about it. But there is a definite age barrier. People who are really older, on one estimate from Kerala data that was done by another IICB scientist on mathematical modeling, it seems the 62 years of age or 60 years of age is the kind of a barrier beyond which the fatality increases too much and severity of the disease also increases too much, right? So naturally, we already know that most of the young people are protected from severe disease. They're not protected from infection. Now, we should not be afraid of infections from happening, right? because we have been infected with so many different infectious agents all through our lives. So that's not a problem. Problem is of the severity of the disease and the deaths happening. Even if you compare with other infectious agents, the deaths, as I said, the case fatality rates are much less. But we are really caught off guard because all the case fatality happened within 7 months. So that's what is actually troubling us. So the understanding should be, the social understanding should be, you have to protect. That should be a reverse isolation of elderly people and vulnerable people. By saying vulnerable or susceptible people, I mean to say all the comorbid conditions. All of us know diabetes and hypertension, chronic kidney diseases, chronic liver diseases. I have seen patients with different cancers undergoing cancer chemotherapy. They're susceptible to more severe disease. So it's better to, because it's very difficult to spread infection. You have seen, I mean, the infections could spread to like all the 7 continents in 2 months. We should not think that all the measures that we are taking is going to prevent infections from happening and will just break the chain, as people were saying. It's not possible. An infection which is spreading by droplet and aerosol, and the infected persons are infective for more than 30 days, it's very difficult to prevent those infections from happening. But you have to do the reverse isolation. But this reverse isolation also needs all the measures we are taking. All the measures that we are taking, social distancing, mask use, avoiding crowded places, these are all actually will help you in this reverse isolation. But we should not be under this misconception that we are actually trying to prevent ourselves from getting infected. Infections will happen if you have to move the economy. Now, because I'm an immunologist, I'm not an epidemiologist or virologist, and this is, I think, a lot of students will attend this webinar. Hopefully, they have already started attending. So I'll talk about something that I know better than virology or epidemiology. I don't know much about it. Now, the immunity of in this host pathogen interaction, I'll just briefly introduce it to you, and it's a very interesting disease. You'll realize after I describe the thing. So all of us know, right? So this audience knows there are circulating white blood cells, which are the major immune cells. The immune cells come in different flavors, and they actually perform different functions. And whenever any pathogen for that matter, gets introduced into the body, so there will be host cells that will be infected, and this infection of host cell causes damage to the host cell that releases some alarming molecules, and these alarming molecules will actually recruit the immune response. Now there are, as I said, immune cells come in different flavors. There will be some cells which do not recognize between different pathogens. It just recognizes that there has been some change in the tissue space. Some tissue cells are getting stressed because of that, so they just react to it. And those cells are like neutrophils, macrophages, and we call it the innate immunity or innate immune response. And there are other cells which have special ability to detect what kind of pathogens they are looking at, which specific pathogens they are looking at, because they have eyes to see them in a proper way. And for example, we call them T cells and B cells, which have the T cell receptors and B cell receptors. And so these receptors can actually recognize specific molecular patterns that can detect, that can help them recognize which beast they are actually dealing with, right? Now, both these arms of immune response are important because the first line defense comes from the innate immune cells, and the second or delayed response comes from the adaptive immune cells like T cells and B cells. Now, when this SARS-CoV-2 infection is happening, what is happening? So we all know now by this time, because these are newspaper reports, right? So ACE2 receptor is docking to the spike proteins on viral surface. It's also using the co-receptor called TMPRSS2, which is an enzyme. The virus enters, replicates inside the lung epithelial cells. Cells die. This picture shows pyroptosis. It may be necrosis or apoptosis. We don't have a very clear data on that. But these cells are dying. And this dying of cells, or these cells getting infected and getting stressed, secrete some of the the biochemicals in the circulation, as you can imagine, and that activates the immune system. Now, once the immune system is activated, what happens? So there are two possibilities, right? In most of the patients, or rather most of the infected persons, it's very difficult to call them patients because a whole lot of people do not even recognize or realize that they're infected. All these asymptomatic infected persons, right? So what's happening there? So again, their lung epithelial cells, they have a healthy immune response. Their lung epithelial cells get activated, or rather stressed. It releases some of these biochemicals. There are innate cells which are getting recruited, which are macrophages and all. There are adaptive cells like T cells and B cells getting recruited, and they kind of control the situation very soon, right? The macrophages engulf all the infected cells. The T cells, CD8 T cells will cause cytotoxic molecules to release. They recognize the viral proteins being expressed on these infected cells. They specifically kill those cells, and very soon the infection is subsiding. But in a small group of patients, what's happening? The immune response is getting induced, and it's induced too much. And there are a whole lot of these cells coming in the tissue spaces. They're producing a whole lot of biochemicals, and then the immune response is actually taking over. Now, the virus is not yet important, or not at all important. Whether how many viruses you have got, how many lung cells are infected, these are not important anymore. Now, what's happening? All these immune cells are producing biochemicals, they are soluble substances, they're moving in circulation. All the immune cells are also getting recruited to other tissue spaces. They're causing a multi-organ damage, right? And this is what is causing the severe disease. So naturally, this is a very unnatural infectious disease. It's not. It's very much unlike a classical infectious disease. Here, how many pathogens are present in the system doesn't matter after a while in some patients where there is an unhealthy immune response or rather hyperactivated immune response. We are hearing these terms called systemic cytokine storms and all, which are actually causing the multiple tissue damage beyond lung. And people, I mean, all this data, all these realizations came from very interesting work that is being done all over the world. This is a very interesting work on single cell sequencing data where I don't want to introduce single cell sequencing. So here what they're doing, they're sending a fluid into the lung and getting the bronchoalveolar lavage. It's almost like washing the lumen of the lung, and to get all the cells out, whatever cells are freely loosely attached to the lung epithelia. There may be lung epithelial cells, and there may be other immune cells which are getting recruited into the lung, and they are actually taking a account of their mRNA expression. Now, when they're doing the mRNA expression on a single cell level, so you are taking one cell, taking their mRNA out and sequencing it to find out what are the mRNAs that are being expressed at a given point of time. Now, these mRNAs can actually give identity of the cell they are coming from, and also the functional status of the cell they're coming from. And naturally, you can figure out what kind of immune response is happening. And most of the explanation, most of the schematic presentation I made comes from all this kind of data, which actually enriched our understanding about the disease biology. But the data that I would try to impress upon you, which is the most important paper that came out from China, who looked into 37 asymptomatic cases who doesn't have any symptom. And what is our understanding about a classical infectious disease? Our classical infectious disease understanding says that, well, if someone's immune response is very robust and the infectious agent comes in, and if the immune response is robust, the infectious agent will not be able to produce disease. And so asymptomatic infections, you would imagine that their immune system is actually working much better. On the other hand, in SARS-CoV-2, this paper actually showed that the immune response is much less robust in the asymptomatic infection. Even the antibody content, the specific anti-SARS-CoV-2 IgG levels are much lower in the asymptomatic carriers. That means you are not really dealing with a classical infectious disease. the disease or all the symptoms and the severity of the disease biology is driven by a hyperactivated immune response. The infectious agent has nothing to do with it. It just triggered an acute inflammatory disease. Okay, so we are actually dealing with an acute inflammatory disease, which was triggered by a virus infection that could trigger the inflammatory disease in a very few number of people whom it infected. A large number of people who are infected, they never get this acute inflammatory disease, which we call COVID-19. Okay, so SARS-CoV-2 infection and COVID-19 is totally different. SARS-CoV-2 infection in most cases doesn't do anything. Now, I'll just try to talk about a little bit what immunologists are trying to do to deal with the situation. Now, immunologists are known for immunization, right? So, immunology as a subject came much later than all these immunological interventions, right? Vaccines came much before any immunology textbook was written. Okay, now, and so there are two ways you can actually protect someone who is getting infected with infectious agent. And so one is you can passively immunize someone, right? So someone is getting infected and their immune response is boosted up or activated, and it is producing different kinds of biochemicals, and you can actually transfer it passively to another individual whose immune response is not that boosted up. Okay, and this is called passive immunization. And all of you, we started this webinar with the discussion on this plasma therapy. Well, plasma therapy is nothing but this one. And you have to remember, this is not something very new, right? This is one of the oldest interventions that immunologists, or rather people who are dealing with immune system at that point of time, they were not called immunologists at that point of time, started devising against different infections. You have to remember, students should know the Emil von Behring got the first medicine Nobel Prize in 1901 for doing this. So he did this convalescent plasma or anti-sera to treat diphtheria infection. So that's why he got Nobel Prize. So we are actually going back to our age-old intervention, immunological intervention, to fight this virus. And this is not the first time we are doing this. Over past 100 years, there has been different pandemics. In all pandemics, when there are these emergency situations, convalescent plasma therapy had been very useful. In some cases, it worked. In some cases, it actually never worked. But nevertheless, people have been going back to this strategy over and over again, just to make a shortcut to induce immune response in someone who is not being able to mount immune response, right? And now you know there are very big trials going on. So China got the epidemic almost 4 months ahead of us. USA got it almost 3 months ahead of us. In USA, there is a plasma therapy trial going on, which is planning to recruit 20,000 people. And very recently, they actually shared their data on 5,000 people, showing that these people never really this therapy never caused any grave adverse effects, and that was again very helpful for all plasma therapy trials all over the world. And so people are now fearlessly moving ahead to get the clinical outcome data. Well, the USA clinical outcome data is not yet out, so very soon we hope that they will be out. In India, there are 10 trials of convalescent plasma therapy going on, which are approved by CDSCO. I'll just mention 2 of them. One is the biggest one, which is driven by Indian Council of Medical Research, which is a multicentric trial and involving more than 50 clinical institutions all over the country, in most parts of the country. And then the trial that I am involved with is a Council of Scientific and Industrial Research sponsored trial along with West Bengal government. Now, there is a basic difference between these 2 trials. In our trial, we are actually looking at, so our primary outcome, our depicted primary outcome is to identify immune correlates for response to plasma therapy. So that tells you that we are already expecting that not all patients will respond favorably to plasma therapy. Some of them will do, some of them will not. And it's very important to know if people who are actually responding to plasma therapy, why they are doing that, and people who are not, why they are not doing that, right? So that kind of data will actually come from the CSIR trial. ICMR trial is more of a clinical trial, looking at the clinical outcomes, so how many deaths are happening, how many disease progression is happening. And so this trial the CSR sponsored West Bengal trial had been the one of the first trials that was proposed. Actually, ICMR trial came much later, and so the West Bengal trial and the Kerala trial started working the very first, and ours is the only one in the Eastern India. So we actually have a good website on this trial, because after we started the trial, we realized it's not a scientific exercise that we are actually took the responsibility of. It became a social awareness program. So we had to. So our biostatistician became a public relations officer, and so because a whole lot of social awareness had to be generated so that you could make people donate plasma after they have after they have recovered from coronavirus infection, because a whole lot of apprehensions were around regarding reinfections and all. We had to motivate them, we had to encourage them, and so things were done. And so this involves a whole lot of activities, like so we contact people who are recovering from patients. We have actually reached out to almost 164 COVID recovered patients, and we could motivate only 30 of them to donate plasma. That tells you that it's almost like a social barrier and social awareness program that we are running along with the scientific part of it. And and we were working amidst the lockdown, right? So naturally, we had to arrange transport from their home to the hospital, where they will be screened, because for any blood transfusion, there are different screenings. For convalescent plasma, definitely you are expecting that this plasma will provide the specific IgG antibody to the recipients. So naturally, you have to test for that. And IICB had been doing all this testing for the screening of donors, and then the actual plasma donation comes, and all these people came forward. They're like heroes in this the really dark times, and most of them are doctors and police officers and nurses. Two students were also there, young students, and so it was very interesting. So I just give some anecdotal accounts. So it's very interesting because it seems people who are fighting. So all these people, most of these people got infected while fighting on the street, right? Like the doctors, like the nurses, the police officers, and it seems that the zeal of fighter remains even after they got infected and they recovered. So they again comes back to continue the fight. So that's really commendable job that they have been doing and helping us in doing this trial. And so this is the 3 institutes that are doing this. So all the plasma donations are done in Medical College Hospital after all the screening part is done. And then this is the product that we come up with, COVID-19 plasma. And then patients are recruited in the IDNBG hospital. So then they give informed consent for going to this trial. And then all the supportive immune monitoring work is done by a dedicated epidemic immune monitoring laboratory at Indian Institute of Chemical Biology. Okay. And while doing these experiments, or while doing this trial, we're also, as I said, we are also looking forward to all the immune correlates of the response to plasma therapy. So naturally, it's also giving us very interesting scientific insights. So I'll just share a few of them. For example, we started looking at patients who are having either mild disease. All of them were hospitalized. We never took samples from asymptomatic infected patients, because it's very difficult to know who are asymptomatic infected patients. But we actually tried to compare the mild disease having patients and the ARDS, means the acute respiratory distress syndrome, which is a more severe form of the disease. And so you can actually look at, so these are just different cytokines that play different roles in the immune response. And you can see the most of the immune response modifier cytokines are actually more expressed or more abundant in case of severe disease. As I was saying, right? So it seems this is a disease, this is more of an inflammatory disorder rather than a host pathogen interaction. So what you expect as an immunologist or an infectious disease biologist? You expect that the mild diseases are happening because the immune system is much more robust, and that's why the disease was not progressing. But in this case, it's the opposite. In mild disease, the immune response is actually showing a very muted response. In the severe disease, it seems the immune activation is much more cytokines are abound, and that's why the disease is happening. And we could actually figure out what are the cytokines that are actually being regulated together, and whether this regulation, this coordinated regulation, is changing when you compare the mild disease versus severe disease, how the network is happening, which are the, for example, we found IL-6 was already reported, we found IL-1 receptor alpha and MCP-1, which is the major chemokine. So these are actually getting like more enhanced in the severe disease. So it's giving a whole lot of scientific insight. The clinical insights I can't share a lot, but very soon we'll get to know about this. Now, what are the other strategies that an immunologist can take? So the next comes active immunization. Now, all of us are really hopeful about the vaccines, and there are almost 150 vaccine candidates being pursued all over the world. But you have to imagine that a trial which is trying to test a vaccine is regulated much more stringent. Because in case of a drug trial, what you do, you have a disease, have a severe disease afflicted patient, and you want to see whether your drug is working or on him or not, right? So very few people are being tested, and also you are actually trying to do a life-saving procedure. But in case of vaccine, what you intend to do, you intend to give a form of the same pathogen which you think that is not going to produce disease, but actually will produce the similar immune response. And you are going to recruit healthy volunteers who doesn't have any disease and give this vaccine to them to see whether they get the infection or not afterwards, right? Naturally, you are making decisions on behalf of a whole lot of healthy population, and whom you are going to give this infectious agent, which may be attenuated, which may be partial, but still, nevertheless, you're doing that, right? And so the regulatory authorities are much more uneasy in approving a vaccine trial rather than other drug trials. So naturally, this is going to take time. So you have been seeing all the projections of 9th September, 15th August, 21st December. Well, these are, please take all of them with a pinch of salt. It's not going to happen like that. In my estimate, it's going to come not before 1.5 years to 2 years, okay? It's not easy. Now, for the students, let us just revisit what are the different strategies that are being taken for vaccine development, which is important, right? And so in vaccine, as I said, what you are trying to do is use either the full components or partial component of the same pathogen, which will you expect that will induce the similar immune response, but will not produce the disease. And so there are different ways you can actually mimic that original pathogen, right? And the most commonly used and most easily developed things are the inactivated or live attenuated vaccines. All of us have taken this kind of vaccines in our universal immunization programs. I'll just point out here. So in the universal immunization program, how many vaccines do you have? Hardly 15, if you consider all over the world. How many infections do you have in our lives? Hundreds. So you have to realize developing vaccine is not that easy, and so it's better to think beyond that. Okay. Now again, coming back to my string of discussion. So inactivated and live attenuated vaccines, we already have examples of that. So many conventional vaccines like influenza, chickenpox, polio, all of these fall into this category. And so a lot of companies and institutes are actually trying to develop SARS-CoV-2 vaccine using this strategy. I know of one effort by Bharat Biotech and CCMB, which are also trying to do this in India. There are DNA vaccines. You supply with the DNA that will express the proteins that are expressed by the virus, and that's how you are planning to induce immune response. Again, we have certain examples of candidate vaccines of this category in some other viruses, but we don't have any approved DNA vaccine as of yet for any infection. Uh, there are vaccines which uses another viral scaffold through which you are going to supply the component of the original pathogen. And so this is the adenoviral vector. For example, you have heard of all of this University of Oxford efforts, and which is ChAdOx, and that's actually using the adenoviral vector. So we have examples of adenoviral vaccines before. For example, the Ebola vaccine, again developed by University of Oxford, the same group, Sarah Gilbert, and they actually were also adenoviral vaccines. There are RNA vaccines, which is being pursued, the strategy which is being pursued by Moderna and in collaboration with NIH. And so they are they have also done the phase 1-2 trial, I think, as far as I know. And there are finally the subunit vaccines, or this is the VLP, viral-like particle vaccine. So you take a liposome and decorate it with the protein or from the original pathogen, which are supposed to induce a similar immune response. And now the liposome looks like a virus, and you give it that. So we already have experience with this kind of vaccine because HPV is a virus-like particle, which is used for cervical cancer protection. Australian effort in Doherty Institute is actually taking this strategy for SARS-CoV-2 vaccine. And then comes the recombinant vaccine or subunit vaccine, where you actually get the components which are supposed to produce immune response, make recombinant versions of it, and inject it to the recipient. But in this case, all the students should remember, in this case, you don't have the other components of the pathogen, and naturally, to induce immune response, you need adjuvants, which are non-specific activators of immune response. All of us have read in textbooks, fronts, adjuvant, alarm, and things like that. And there are definite recombinant vaccines available. All of us take it. So we have a good experience with the recombinant vaccines. The major is hepatitis B vaccine, right? And so now usually we are really afraid of vaccines, and now we are really looking forward to having a vaccine soon enough. But unfortunately, I don't think it will be that soon that we are going to get a vaccine. And so New York Times did a very nice analysis in late April. I like it very much. I cite it everywhere. So they took our experience of vaccine development over past 50 years and then tried to map it to the timeline, right? How long a vaccine should take. And based on human experience of vaccine development over past 50 years, SARS-CoV-2 vaccine, according to these calculations, should not come before 2036. Okay. And then they said, okay, this is a unprecedented event. A pandemic is going on. People are really crazy about getting a vaccine soon enough. So you can actually squeeze few steps to a very short time point. And we are already seeing it. For example, you can actually squeeze the academic research and preclinical and phase 1 trials almost running parallel. And we actually saw that with the University of Oxford efforts. But still, the next steps are the major time-taking steps, the phase 2 trial, phase 3 trial. And then you have to build factories. You have to generate 7 billion doses to protect the world's population. So the manufacturing will be there. Now, when you're preparing a vaccine for a trial, how many doses you are preparing? You are preparing 100 doses, at best 1,000 doses. When you are generating billions of doses, there will be batch-to-batch variation. And you have to first find out whether there are batch-to-batch variation. Then you have to find out how to standardize the batch-to-batch variation. Then you have to find out how you can minimize the batch-to-batch variation. So it involves a whole lot of things. So according to their estimate, again, it's a very pessimistic estimate. Their estimate is you can hardly bring it down to 2032. I'm pretty sure if this is the timeline we are looking forward to, then coronavirus will not be a problem anymore at that time point. But I'm not being that pessimistic. The people will, I think, squeeze all the steps given the emergency, but still it's not going to come before 2 years. Then what is in your hand? So preventing the the infection using a medical agent is not going to happen soon. So what is in your hand? You have to save the people who are getting severe disease. Very few people are getting severe disease. Almost 80%, current estimates are 50 to 80% of the infected persons are showing any symptom. Among the people who are showing symptoms, only 5% are critical. 15% are having severity of disease to the extent that they need oxygen support, right? So you have to develop drugs and strategies to save those people who are actually having severe disease. Then this mayhem is not going to be of that much of a problem. You have to understand. Now, again, what about the drugs? So what are the drugs that are trying to develop? Now, there is no time to develop a new drug against this virus, right? The pandemic is ongoing, 18 million people infected within 7 months. So there is no time to take a conventional drug development program, coming up with a drug that will be specific for SARS-CoV. Come on, it's not going to happen. So what people are doing all over the world, they are trying to repurpose existing scaffolds which are approved for human use. That saves your time. And there have been many candidates, all of you know about this. They are actually targeting different phases of the natural history of the disease or the infection cycle of the virus. So you can actually target the ACE2 spike interaction, which will be targeted by the monoclonal antibodies, and the convalescent plasma is also supposed to do that. You can target the TMPRSS2 co-receptor, which is, there are agents for that, chemostat and nafamostat. People have been using chloroquine and hydroxychloroquine, much talked about drugs. So they have been shown to prevent the endocytosis of the virus inside the cell. There are virus RNA polymerase inhibitors like remdesivir, which is RDRP inhibitor, right? RNA-dependent RNA polymerase inhibitor. And so these are being tried for. Now, again, I'll go back to the realization that we got from the asymptomatic data. What was the realization? The realization was this is not a classical infectious disease. It seems the infectious agent is not wreaking havoc. Rather, the immune response it triggers is wreaking havoc. All the drug trials, I'll cite 2 major drug trials that also supports this view. So this is a trial with remdesivir, okay? Published in NEJM, very high profile trial. Now just look at the data. So remdesivir shows some effect. Still, it's not very strong. Shows some effect when you are giving it early, when still the virus is replicating and the initial immune response is being generated. But later on, after the disease has progressed to the stage wherein all the untoward outcomes are being resulted in, remdesivir has no role to play. So antiviral doesn't have any role in a viral disease. That tells you this is a very non-classical viral disease that we are dealing with. Okay, now go to the next drug, which is dexamethasone. What is it? So this is a corticosteroid. This is an immunosuppressant, immunomodulator. Again, we all know about this recovery trial data, but you should take a closer look at the recovery trial data. It again confirms that we are actually dealing with acute inflammatory disease. Look at the data. The first you look at this one, okay? So here you are actually, when you give steroids to milder patients or very mild disease, dexamethasone is actually worsening the condition, right? Why is it doing that? Because just remember the schematic that I showed. A healthy immune response is being generated, which is getting rid of the virus infection, okay? And now if you give dexamethasone, it's not going to work out that way. Now, just look at the more severe disease conditions. People who are needing oxygen, people who are needing mechanical ventilation. Then the virus has no role to play. And this is what is happening. This is the schematic representation of the immune response. Here is a dysregulated, hyperactivated immune response that is happening. And now dexamethasone starts showing its effect. And these are the patients who are going to have untoward outcomes. I'm telling you, these patients are mostly recovering by themselves. Don't have to do anything. A mild symptomatic therapy is actually doing the wonders. For these patients, we don't yet know how to save them. Some of them, I mean, again, a lot of these patients are also going back after getting oxygen support for a few weeks, but a lot of people are not going back, and there are untoward outcomes. And for these, it seems immunosuppression is going to work, right? Now, I'll just end by mentioning a little bit about herd immunity. This is something that I have been asked a whole lot of times by media and also close friends. And because they think that I understand this thing, I don't think I understand. I'll tell you. So, what is herd immunity? Herd immunity is herd immunity calculations are all based on vaccination programs. Okay, and when all the population are susceptible and the infection starts and no one is vaccinated, that means no one is protected. The infection spreads like this, right? It reaches all of you. And then if you get 20% people who are protected by vaccination or by natural infection, the spread is little bit slower. Some people actually are spared. Now, if you go to 40%, the spread is much slower, right? And at the end of the epidemic, you actually see a whole lot of people are spared. And it goes like this: 60% vaccinated, it is much less speed, and much more people are spared. 70% vaccinated, again, even slower, and things like that. So this is what is herd immunity. And the sheer speed this virus is spreading with, you can rest assured, herd immunity or a spread of infection to a level where a whole lot of people are protected because their chance of getting in contact with an infected person is much less compared to a protected person. So that will come much earlier than a vaccine. I believe so. So all this herd immunity discussion, initially the herd immunity discussions were absolutely no, right, in public. So people were really scared about talking about this because a whole lot of people were dying, still they are dying, and the deaths were happening within a very short time. But soon enough, even if we didn't like it to discuss it or not, soon enough we realized the infection spread is too high, right? All the serosurveillance data started coming in, and all the serology kits which are detecting past infection now. So your RT-PCR will actually detect active infection, and then after recovery for a few days or few weeks, it can be positive. But the serology will actually detect for several months. And at this point, there are data that the antibody levels are coming down, which the serology kits are checking after 2 to 3 months. But again, that doesn't mean that there is a waning of protective immunity, right? No way there is protective immunity. This is the data from different parts of the world. How many people are actually seropositive through the serosurveillance efforts. In Mumbai, you can see in Dharavi, 57.8% people are infected. Non-slum areas, 16%. In New York, there are 22.7% in New York City. Okay, so that tells you that all the densely populated areas are getting spread of infection really fast. And in a calculation, mathematical models, etc., it seems like 60 to 70% people have to get infected to get a herd immunity achieved. But in practical terms, it's not going to be that higher percent because a whole lot of people are already protected. For example, kids are contracting much less infection, so almost 25% of the population is gone. And also in all these mathematical models, all the people are taken to be equally susceptible to contract infections, which is not true in human society, right? My mother is much less susceptible to contract infection than myself, right? So there are gradations of susceptibility, gradations of chance of getting infection. So naturally, even much lesser percent will actually give you herd immunity. But still, we can't really go for herd immunity and lifting the lockdown. Why? Because our health infrastructure is not ready to accept so many infected people within such a short time. Because this has been unprecedented. No other infection have registered 18 million infections within 7 months. No other infection has registered 700,000 deaths within 7 months. No country is prepared, their health infrastructure is prepared to take this, right? So that's why you are trying to slow down the spread of infection so that your hospitals are not overwhelmed or your health infrastructure is not overwhelmed, right? And so I'll just end by saying that we have to buckle up for a rough ride because vaccines are not really coming very soon. And so we have to get the numbers on our side in terms of how many people are infected but not having severe disease and their recovery. And just to tell you, I mean, this is there is a like atmosphere of fear all around. But you have to remember, 18 million people have contracted infection. It's true. Again, 11 million people have recovered from the infection without any untoward aftermaths. So we have to be really, I mean, we should pay heed to these facts also, and we should not be that afraid and that apprehensive. So I'll be happy to take questions if from the audience. So I think I'm done with this. Thank you so much.
Thank you, Dr. Ganguly, for your nice presentation. Questions have already started coming. I have a question from Priyam Das. He says that he is COVID positive at present. He is a chemistry student at IIT Guwahati, and he has got some specific questions. Like he says, if the immune response aggravates matter, should I stop taking immunity boosters? That's
No,
the first question.
It's not about that. So immunity booster, I don't know if there are specific components or specific elements that can really boost your immunity, and if it does, how it does so, except for a few of the vitamins. And so this is not about not mounting immune response. As I said, the mild disease, there is also mount of immune response. That's how the virus is not being shed, and after a few weeks, usually 2 weeks, people are recovering, right? So immune response is required. The problem is happening with the dysregulated immune response that is happening in a whole lot of people, and that's where the problem lies. And that is also happening in anti-inflammatory, like inflammatory diseases, autoimmune diseases. So because there are autoimmune diseases in human medicine, that doesn't mean that you are not going to have immune response, right? Immune system has to be there. A healthy immune system has to be there. Still, people have autoimmune diseases, but you have to deal with the severe manifestations of this disease in that light. That you are actually dealing with an inflammatory disease, not an infectious disease. That was my point.
Okay, here's a second question. I have done my second test after 15 days of quarantine and asymptomatic, but still it is positive. When is it the suitable time for the next one? And there's a third one, if I can put it right now. How long do I hold the immunity and chemical units that prevent me from a second infection?
Well, second infection means reinfection, right?
Okay.
Well, first thing is second RT-PCR. Well, RT-PCR will remain positive for really long. Okay, so this is the problem. That's all this. So your first question is related to the second question. So it has been a scare in actually West Bengal. Last week there were some reports reinfection is happening. So this is just because the testing method is RT-PCR. And RT-PCR is very there are chances of false negatives are really high, right? There are chances of false negative. So a whole lot of people who are first positive, then the second test they are negative because they were false negative, and then they are coming back with false positive. I mean, the true positive, and then they think that they recovered, and then they're again getting infection. No, there is no report of reinfection all over the world. There have been 18 million infected cases from all over the world. If there were considerable number of reinfections, you wouldn't have missed it. Okay, period. How long the protective immunity lasts? The pandemic has been there for past 7 months. In past 7 months, there had been no report of reinfection from anywhere in the world. out of 18 million infections. That tells you that the protective immunity is for 7 months. If I had this webinar 3 months from now, and still there were no report of reinfection, I would have said the protective immunity lasts for 10 months, because there is no other way to learn how long the protective immunity lasts unless you have experience of reinfections. Okay, there is no scientific method to predict how long the protective immunity is lasting.
Okay.
Here the only experiment is time. Okay, so for you, I think in another 2 weeks you should check it. You should not worry about it. Usually the positivity, RT-PCR positivity remains for long, but you are not infective after 14 to 15 days. That's the in general data, but there are individual to individual variations, but you need not to worry about that.
Okay, so there's another question from Dr. Amun Mondul. He says dexamethasone is immunosuppressive. So to control SARS-CoV-2 with the dexamethasone, patients are vulnerable to other infections.
Yeah,
How
of
do you
course.
justify?
Yeah, of course. But you are saving that patient from dying, right? Of course, dexamethasone will do a systemic immune suppression, and these people will be susceptible to other infections. But you can imagine all the severe disease patients will be admitted to ICUs, and hospital care will be given, and so other infection chances will be less. But to save them, dexamethasone seems to be working, at least in recovery trial. So all the trials are also going on in our countries also, and so it has been part of the general protocol. Yeah, it's accepted. You increase the chance of other infections, but there is no other way.
Okay, so there's a question about vaccine which I received earlier in the registration form. Can one vaccine be done within 6 months, like the recently discovered Covaxin or the Chadox one?
Cannot be done.
Okay.
Impossible.
Okay, so there's another question I find, what is herd immunity and can it be effective to prevent or resist COVID-19? I think you have already answered that, but still, since there's the question, can you please take it?
Yeah, so herd immunity will be reached at some point of time, whether we want it or not, right? But the problem is, say you attain herd immunity after 10 months. And your protective immunity lasts for 10 months. So what will happen? So there will be repeated epidemics, right? So at this point of time, we don't have a very clear idea, even if herd immunity is achieved, what will happen. Okay. The only thing I said is there will be a sheer spread of infection reaching numbers which are required for herd immunity even before vaccine is there. But we don't know the vaccine candidates, how long the protective immunity lasts. They don't have a data. We don't know how long the natural infection protective immunity lasts. We just know 7 months. And say the herd immunity is achieved in 15 months and your protective immunity lasts for 12 months. What will happen? So these are unanswered questions, and there is no scientist, no doctor can give you this answer. These answers will only come from timely experiences. Okay.
Okay, so there's another question regarding herd immunity. When can herd immunity be achieved?
Yeah, we don't know. But with the speed of infection, I don't think. So again, herd immunity, whether it will be achieved, we don't know. We know the percent infection of population, which is usually thought to be helpful for herd immunity or helpful for achieving herd immunity, will be reached soon enough. But whether that will give you herd immunity, we don't know, because we don't know how long the protective immunity lasts. So these calculations are based on an assumption that the protective immunity lasts lifelong. Okay. We don't have that data.
Okay, there has been many questions regarding diet. Is there any specific diet for COVID-19 patients or any dietary substance which can boost your immunity? They are being said to be immunity boosters. There's been from the Ayush, they have suggested taking certain things, herbal products. which are immunity boosters. So, what do you think about that?
Well, I'm not an expert, so I don't want to comment on that. A nutritionist will be better positioned to talk about that, right? I don't have much knowledge on this.
Okay. There's another question. Is there any vaccines available for hepatitis B in India?
Yeah,
is
of
it
course.
curable?
Yeah.
And
Hepatitis
it is
B, we have recombinant vaccine. Yes.
Okay. Since no other question have come up in this over here, I've got questions from people have asked. What it would be interesting to understand the mechanistic role of COVID virus towards triggering lung disease or heart failure. Can you throw some light on it?
Yeah, so people have seen the heart failure, left heart failure, and because you are actually having an acute respiratory distress syndrome, so there is resistance in the pulmonary bed where oxygenation happens between the alveolar air and the blood circulation, right? And so if there is a major resistance of oxygenation, the heart will be able to, heart will face greater resistance because it has to pump against the lung resistance, right? So heart sends the blood to lung, and naturally there will be right heart failure. And the left heart actually pumps to the systemic circulation, so its resistance is from the systemic vasculature, right? And so we perhaps will know to some extent what are the mechanisms ARDS is happening. It's very difficult to explain it in here, but because the lung epithelial lining is getting damaged and there are matrix changes in the extracellular matrix of lung, and that's why there is greater resistance and less oxygenation of the blood, and that's how it's happening. And there are reports, perhaps you also know, there are reports of intravascular embolism or thrombosis, and that what happens if there are capillary vessel beds which are blocked because of coagulation defects. What will happen that some of the alveoli will be excluded from your perfusion or from your oxygenation bed, right? So because blood is not reaching those alveoli, even if oxygen is there, blood is not reaching. So the surface area of oxygenation reduces. So that's another cause of the respiratory failure, usually the type 1 respiratory failure. Yeah. So there are some understanding, more understanding is needed, definitely.
Okay. So there's another question. Drug development or vaccine development? What is the better strategy in the control of COVID-19?
Well, good thing is the same persons are not going to do that. So no harm letting drug developers develop drugs, getting vaccinologists develop vaccine, right? But at this phase of the pandemic, I would say you need drugs. You need drugs to save severe disease patients. You need to save people from dying. Thinking about preventing infection is not going to help you. Those people are dying, right? So you need drugs to prevent death in the severe disease.
Okay. There's a question. Those patients who are on immunosuppressive drugs, what to do with them in relation to protection and treatment?
Well, as I said, all the susceptible individuals have to be reverse isolated. So you have to take certain measures so that they are not contracting or getting in touch with people who are moving around, because people have to move around and there is no harm. You have to move around, run the economy, gain some infections to help herd immunity, I would say. But those people who are more vulnerable, more susceptible, older age, please isolate them. So there is no medical method or methodology to prevent them from getting infected other than just isolating them. So please do.
Okay. So there's another question. Can you please tell us that whether any particular cytokine out of the many has been noticed to get triggered during this vivid infection, and which is the most responsible for the comorbidities?
Yeah, it seems interleukin 6 is a major thing, because there are trials which are using antibodies against IL-6 receptor, which is called tocilizumab. and people have shown, like corticosteroid, tocilizumab has a very good effect in these severe disease individuals, and it has been a common practice even in Kolkata and even in Indian medical practice in COVID-19 that they measure IL-6. A whole lot of severe disease patients actually show heightened IL-6 levels, and then tocilizumab is used, and usually they respond to the tocilizumab therapy. Yeah, IL-6 is the major thing. I think there are a few other cytokines that people will report soon.
Okay, a question has come up. I think you have answered, but still, is there any possibility of getting infected for the second time? Suppose
Yeah, like
he
I
is
said,
cured.
yeah. I mean, so we don't know. So at this point, we can say that within 7 months, you are not going to get reinfected.
Okay. Then there has been a question that since the virus utilizes ACE2 receptor to get entry into the cell, so any pharmacological drug against this ACE2, can it prevent the infection? When I just close the door for the virus, it won't enter.
Yeah, theoretically, yes. So theoretically, yes, people are actually trying to repurpose drugs based on computational algorithms to find out which can block the spike ACE2 interaction. And also all the neutralizing antibodies actually work through that. So they don't target ACE2, they actually target the spike protein. And that's how it interferes with the interaction between spike and ACE2. So definitely that can be a strategy, but we don't know of any drug at this point that can specifically prevent spike ACE2 interaction. Yeah, people are trying to find out.
Okay, so there's been a question that coronavirus 2, what is the proper incubation time of it in the body?
Well, there are varying estimates, and you I mean, that's not unexpected, right? So human to human variations are there, and it seems like 4 to 6 days is the incubation period at this point. That's the median. So mean is not going to help you in this kind of cases. Median is the one that. So what is the maximum number of people experiencing? So it seems it's 4 to 6 days after you contract the infection and getting first symptoms, if you at all get any symptoms. Okay, if you are not getting symptoms, that's why it's very difficult to calculate. A lot of people are not getting symptoms.
Okay, so there's another question which asks, let me check, if so many vaccines are there, candidate vaccines, but the virus is also mutating. So will these vaccines work in the long run?
Yes, there is a theoretical possibility of vaccines being effective against certain virus and not other viruses. But I can tell you as I said, the humans were much more prepared this time, and whole lot of viruses were sequenced from different countries. And even in India, almost 1700 viral isolates have been sequenced already. Even more than that, I believe. TBT registered almost 1,000. CSR did almost 1700. So from those data, it seems there are mutations because our RNA virus is susceptible to faster mutation rates, right? Because RDRP, or RNA-dependent RNA polymerase, doesn't have a proofreading activity. So that's why the random mutation acquisition will be much higher in RNA virus. So having said that, the mutations that have been registered, it's a really fast mutating virus, but the mutations that have been registered till date doesn't change doesn't seem to change the virus to an extent that it can be said this is a separate viral strain. So all the different mutations are still at the clade level, and they don't really change the character of the virus to an extent where the immune response is getting changed. Okay, so your vaccine will be vaccine efforts will be jeopardized if a virus acquires a mutation that changes the immune response. So then your vaccine will be difficult to universalize. And at this point, we don't have such an evidence. But having said that, this is an RNA virus. It's mutating really fast. it may well happen that the very next mutation that is registered is changing the virus to an extent that the immune response is changed, right? No one can say that because mutations are acquired randomly. But till now, we haven't got mutations that can differentiate the viral isolates into different strains. They are only at the level of different clades. Some proteins are acquiring some mutations like.
Okay, so another question has come up in the YouTube chat box. If plasma therapy fails for some patients, is there any chance to trigger the infection by antibody-dependent enhancement?
No, so yeah, ADE was a definite theoretical possibility because in some other viruses we have seen similar things to happen. But I can tell you there have been specific directed experiments towards that, and there is no evidence of ADE being happening, at least in vitro, because ADE, if happens with certain antibodies, usually can be registered in vitro. And in this virus, we don't think there are chances of ADE at this point, as of now. Yeah.
Okay, so there is a question: what is the easiest process to make vaccines against COVID-19?
Well, it depends. I mean, I'm not a vaccinologist, but I can tell you, like if I just go to your position and look at all the strategies, methodologically, I think the inactivated vaccine is the easiest, right? But the problem is the easiest may not work, and so there lies this problem, right? So easiest may not work, so you have to approach from different ends. So that's why among the 150 candidates, a whole lot of them are inactivated vaccines because that's easier. Sub-unit vaccines, or in other words, recombinant vaccines, are also perhaps easier than other ones. But you don't know which will work, which will be giving you the desired immune response that can protect someone from natural infection. So we don't know.
Okay, so we are perhaps running short of time, but still, if you can manage some questions. There has been a question from the registration form. What is the difference of this SARS-CoV coronavirus 2 from other RNA viruses?
Again, I mean, I don't know personally. I am not an expert in the virology. But there are definite homology data with different RNA viruses. But I can tell you that the homology doesn't say anything about biology. Okay, so I always believe that. And so there may be greatly homologous proteins performing greatly different functions. And naturally, your question is actually saying how the proteins of this virus is different from other RNA viruses. And so it's very difficult to say. I mean, this is very stochastic process. Your homology doesn't say anything about biology. And definitely we are seeing that it's much more contagious. There is definite changes in its biology. That's why an infected person is keeping on shedding virus for almost 30 days. There have been registered super infectors, super spreaders who have been infecting or shedding viruses for almost 50 days. And so this definitely this is host biology as well as viral biology. And there are other RNA viruses which stay in, where persons stay infected for 5 days. So that tells you that there are variations which cannot be predicted from the protein structure or homology or things like that. Okay. So there are other things we don't know. Yeah.
Okay. When subjects are suffering from ARDS, do breathing exercises are beneficial? Are breathing exercises beneficial for them to overcome their physiological conditions?
Well, I am not an expert, so I don't want to comment on this. Definitely, more expert recommendations will be important in this kind of things. Okay, I think breathing exercise, you mean to say pranayama and things like that.
Right.
I'm not very sure of the biology of it, and so there are other experts. Please contact them because I don't want to comment on this, which I don't know.
There's a question. Sometimes anti-sera testing shows negative results, but RT-PCR shows positive result. Why?
Well, there are 2 points, right? So usually it should not happen because in RT-PCR, false positives are rare. Okay, because you're doing PCR with specific primers. So false positives are really rare. False negatives may happen because your PCR reaction may not work. All of us who work in biology, we know that, right? A PCR reaction doesn't work. That doesn't mean that you didn't have the template. You had the template, but the reaction didn't work. So false negatives are really common. So the major reason that may happen is that your antibody production window has not started, right? So you are positive for the virus, but the IgG, because for antibody kits, you can only know about the presence of an antibody for which your kit has been developed. So usually the kits are developed based on a specific antigen. So maybe you are looking at with an antibody kit which is looking for an antibody which is not present in your body, but your immune system responded with another antibody. Say your kit is looking at anti-spike IgG, and your body responded with an anti-nucleocapsid IgG. Then you're not going to get anti-spike antibody in your body, right? This is one point. Second and first point, as I said, maybe your antibody production has not started yet, has not ramped up yet. That can be detected by the kits.
Okay, Chiranjit, do we have time? Can we take a few more questions? Chiranjit or Shwandip or anyone?
Yes.
Okay,
I think we have 5 minutes.
But he can allow me, please, if you can allow. I just have some questions right now. The question is, can the cytokine released in COVID-19 patient's body affect the patient's cognition?
We don't know. I mean, I'm pretty sure there will be a whole lot of literature that will be coming on post-corona syndromes. There will be aftermaths of corona, right? For example, as a student of medical science, I mean, just not an expert, as a student of medical science, I'm really interested to know all these patients who are having 15-day long oxygen therapy, 100% oxygen being delivered to their body for 15 to 20 days, and then they are recovering. What this oxygen is doing other than saving them, right? So there will be different reports afterwards. I mean, how the corona recovery also left some scars in the body, and the possibility that you said may happen, definitely may happen. I don't know, we don't have data as of yet, but there will be definitely aftermaths of this infection, because this is a almost very long standing infection. People are getting recovered after 20 days. Naturally, there will be scarring, some kind of scarring happening to the organs that they are affecting.
Okay, the next question quickly is, vaccination safe for all kinds of patients, like heart patients?
We don't know. We don't have a vaccine, so I don't know. So
now, if we have a vaccine, can a heart patient be vaccinated? But the question is like that.
If we have a vaccine, you have to make sure that they are not toxic to any group of patients, right?
Okay. So, can we take vitamin C rigorously?
Yeah, you can do that. No harm. I don't know whether.
What other vitamins would you suggest that we take right now? But as an immune booster,
you should not have a vitamin deficiency when there is a pandemic happening all over the world. That's my point, right?
Okay.
So definitely you should take nutritious foods and be healthy and satiate your hunger. That's it. Okay. So don't get malnourished at a point when the pandemic is happening. That will be really a bad thing to do, I'm telling you.
Okay. There's a question over here. Does lopinavir and ritonavir combination help in any treatment as drugs? It would be helpful if you could throw light on it, and because my medical prescription advises me the same, but the side effect seems problematic. This is the same person who was COVID positive and asked you the first question.
Yeah, I mean, I think there are some positive results from few trials of lopinavir, ritonavir use, and your doctor has advised this, and I think you should abide by his advice. And regarding the toxicity, I'll just remind you. So in many cases where infection is for 15 days at most, you are taking the drug for 15-20 days. Perhaps the toxicity, even if it's there, is not going to affect you that much because you are taking it for 15 days, right? So this kind of toxicity becomes really important for chronic viral infections. For example, HIV. A person is taking this these antivirals for long, and then the toxicity becomes a real problem. For this kind of infection, I would say just abide by clinicians' advice. Toxicity is not something that you should be very worried of.
Is there any chance of spreading this virus through breast milk?
I don't know whether there is any data, but that will be definitely a good data to have. I don't know whether someone has looked into it. Yeah, I mean, even fetal-maternal transmission, I don't think there is any data at this point. But these are really important data that will be required to manage this problem. Yeah.
Okay. Cytokine storm is one of the important factors for deterioration of the patient. Is there any difference of cytokine storm in comorbid patient or normal infected person?
That's a very interesting question. Actually, in our work, we are actually trying to look at that whether at least metabolic comorbidity is showing different kind of immune response. We don't have that data from anywhere in the world. I think a whole lot of people are trying to find out, but this is a very interesting question, very important question. Yes, we don't know. The answer is we don't know. We know that there are some core modules which are getting activated in irrespective of whether there is comorbidity or not. But whether metabolic comorbidity or a hypertensive comorbidity or a renal comorbidity, they will have a different kind of immune response, which these data are really important to get, but we don't have that data as of yet. Yep.
Vaccines from different countries shows what type of differences? Do they show differences?
We don't have any data. I mean, so only phase 1 data has been generated. Phase 1 means few volunteers have been given this vaccine and they showed that there is an immune response against the vaccine and they are not toxic. That's it. That's what it says in phase 1. So we don't have this kind of data.
So no use just waiting for the Chadox one coming and helping us out.
Well, time will tell because they have to generate a whole lot of data. They have to find out. They just got, say, I don't know how many, I think 100 patients, 100 individuals they have injected with this vaccine. They just reported that it's producing immune response up to 50 days. Okay. Now the question is whether this immune response is good enough to prevent natural infection from happening, right? So those kind of data will come in the phase 2-3 trials, right? Now the phase 3 trial will tell you all these things. Then there will be phase 4 trial where different ethnicities will be given to the vaccine, and then you will find whether it actually can prevent infection in a large majority of the population. So whole lot of steps are remaining, and the remaining steps are much more longer than the phase 1, right? So yeah, so let's wait for the real data to come.
Okay, what is the mechanism of immune amnesia?
Okay, that's an interesting question. Yes, so immune amnesia. I don't know if he's a student or faculty.
She's a faculty. She's a faculty at the University of Calcutta, Dr. Pubali Thor.
Okay, yeah, that's a very important question because this is an RNA virus. So I think you are insinuating that whether in SARS-CoV-2 there will be immune amnesia or not, right? Now, immune amnesia has only been reported in case of, as far as I know, in measles. There was a very high-profile paper in Science last year where they showed that in measles virus, the previous B-cell memory is being erased. Previous B-cell memory is erased for a lot of other infections. That was a major paper, and that was a very interesting paper. We don't know. Now, measles is also RNA virus. SARS-CoV-2 is also an RNA virus. At least the toll-like receptors or the innate receptors that will respond to measles virus are same with SARS-CoV-2 virus. But there are so many other RNA viruses where Immune amnesia has not been reported, right? So we don't know. But this will be certainly a very important thing to look at, whether immune amnesia affects SARS-CoV, is affected by SARS-CoV. Yeah.
Okay. Can we silence SE2 gene by siRNA or any molecular seizure? And could there be an adverse effects targeting the SE2 as it is involved in several physiological processes in humans?
You can't do that. Yeah, you can't do that. This is too important for us, for our existence.
Okay. Okay. So I'll take two, I think, philosophical questions rather. How mental stress, especially the fear of COVID-19, can affect the immune system of our body?
Well, this is not a physiological, this is not a philosophical question. This is actually a physiological question. There are hardcore data coming in from psychoneuroimmunology, right? So there are definite relationship between the immune response and the psychological stress. Well, mechanistically, we don't know much, but we know that there are vagal innervations, which are the parasympathetic system, and there are vagal innervations of bone marrow, there are vagal innervations of spleen and different lymph nodes, and vagus nerve is highly affected by psychological stress. And if there are vagal instructions to immune system, you can imagine that there will be psychological stress affecting immune system. So definitely you need to, yes, psychological stress, I think it has been shown in some good papers, mechanistically strong papers, to affect immunosuppression. So definitely being psychologically stressed is not going to help you during a pandemic.
I've been informed by a technical team that we are running short of time, so I will just take the last question. The last one is panic of COVID much more than reality?
No, I mean, so there is no scale, right? There is no tangible scale that tells you what is the reality and how much panic you should how much panic you should get. There is no scale, defined scale. People panicked and are panicking because of the sheer speed of spread. 18 million infections happening in 7 months is unprecedented. 700,000 deaths happening in 7 months is unprecedented, and that makes you forget that the infection mortality is too low. Okay, so you are seeing so many deaths happening all around, but you forget that so many people are infected all around. So you don't really tally these two numbers. If you tally these two numbers, then the fear will be less. But that you can't really blame them because it's unprecedented. This kind of speed of infection spread and accumulation of death is unprecedented in human history. So naturally people will get panic. But I would suggest, yes, not many people are getting severe disease. Even people who are getting severe disease, very few are having untoward outcomes. So we should not get that fearful also.
Okay. It seems we have to end here today. Okay. I express my heartfelt gratitude to Dr. Dipyaman Ganguly for accepting our invitation on behalf of the Department of Physiology and Raja Peryman College on the whole. And I would just like to make one announcement rather, for the participants. Please fill in the feedback form, which will be sent to you within 7 days. Once you fill it up and fill it up correctly with your affiliations, you will get your e-certificates. Now I request Dr. Krishnanko Shekhar Gayan, our Secretary of the Teachers Council of Raja Peryman College and IQAC member, to formally deliver the vote of thanks. Over to Dr. Gayan. Dr. Gayan, please unmute yourself. Krishnanko, please unmute yourself. Krishnanko, please unmute yourself.
Is it now audible to all?
Yeah, it's audible.
Okay. Actually, there was a problem with my internet. Okay. On behalf of the Department of Physiology and IQAC of Raja Peary College, I would like to thank our honorable speaker, Dr. Dipyaman Ganguly, for an interesting as well as very informative lecture. From this webinar, we got a clear idea about the immune system and immune response of our body against viruses. Thanks to all the participants for their active participation in this webinar. Thank you to our Microsoft support team, namely Dr. Chiranjit Mondol, Professor Sandipan Sarkar, Dr. Prabhat Lama, Professor Ovik Kumar Basu. Without their support and cooperation, we could not arrange this webinar. Last but not the least, I would like to thank our principal sir and faculties of Physiology Department and IQAC coordinator for arranging this type of webinar. Thank you all. Have a nice evening. Thank you.